Hello. Yeah, that's nice.
I can't see myself anymore in these pictures.
I see. I still didn't do my hair this morning. Well, doesn't matter. Okay. Welcome everyone. Hope you had a good lunch. So we stopped at 12:15. When did the other group stop?
About the same.
Yeah.
Yeah.
Yeah, we didn't see all the movies, but we skipped a few, but we got the essential ones. And I hope that Katrin and Daniel and Maria didn't explain too much. So there's still something new in this lecture now. Yeah, I'm going to stop this video here. Okay. So everyone has a coffee?
Yes.
Yes, good. Ah, Marcus, you're still there. Okay. Okay. Let me, start sharing. Ah, Gustavo. Okay, share screen. Okay. Okay, so in the next 45 minutes, we go a little bit through what we did this morning to get some sense of what's going on. Early embryogenesis. So the question is, how do you get from an egg to a formed embryo that has basically all the features that it can live, survive, grow, and prosper? We work with model organisms, so that is pretty obvious that, hold on a sec. Why is this not working? Okay. You have an egg, you have an embryo, and in between you have embryogenesis. What is remarkable here is that when you start with an egg, these eggs are highly specialized structures. You know, if you live in an aquatic environment, it's different from living in a dry, arid environment. That's why you have different eggs in xenopus and shark. In mammals, the embryo develops in the placenta. In utero, so embryos are very small. There is not really much of an egg to see, just an oocyte. And in zebrafish, it's a bit like xenopus. But as you heard already this morning, I think that, for example, early development cleavage and blastula is a bit different from xenopus to zebrafish, especially when it comes to these cleavage divisions in the beginning, which are a bit different. Nevertheless, although these embryos look very different in the beginning, all these vertebrates go through something that's called a phylotypic state. And I have this. And what is meant by that is that the phylotypic stage represents all the basic features of that clade, which means they have a central nervous system that you can see. You know, they have the basic features like here in the xenopus, you see the otic placode. Also, the fish has that. I don't know why I have these. Let me check do something here. Oops. Never mind. So this is a very typical. So they have somites, they have a null tube, they have the eyes, they have lymph buds. So it's very similar. And during this phase is when these Hox genes are being expressed and do their job in regionalizing the embryos. And in the end, of course, a chick looks very different from a fish. But the fact that these embryos are very similar during a certain stage of development means that the basic systems are conserved in all the vertebrates. So what we did this morning is we looked at quite a number of embryos, the videos that Jan Min had taken, and we looked at the basic features. So we saw that early on, for the first 2 hours, you have these cleavage divisions where you have an incomplete cell division. So that cells remain attached to the yolk. You can inject something into the yolk and it will be transported or diffused simply into the overlying embryo. So these are called superficial cell divisions. In Xenopus, you don't have these. They are complete cell divisions. And then later on, you have the blastula. So you have a mass of cell, and at about 2.5 hours, you have these 1,000 cells. Or was it 3 hours? I don't know anymore. So that means you have 10 cell divisions generating 1,000 cells within 20 to 30 minutes between each cell division. So that means they divide as fast as E. coli under perfect conditions. So that means there is no interphase, there's no G2s. You go through S phase and mitosis. So that is quite rapid, and that is possible because of the maternal contribution. We talk a little bit about maternal contribution at the very end of this lecture. The third phase in development early is gastrulation. Gastrulation means it's the formation of the gut in a way, but what we mean as developmental biologists, it's really the formation of germ layers. So you have 3 germ layers, the ectoderm, the mesoderm, the endoderm. In fish, we don't really differentiate much between mesoderm and endoderm. We call it just mesendoderm because it's a mixed population of cells early on. So when I say mesoderm, I also mean endoderm. So mesendoderm, I just call mesoderm because it makes it easier for me to say. Okay, so what was important early on, you have this differentiation in A, you have the embryo, the cytoplasm on top, and the yolk at the bottom. So that constitutes already a body axis. It's the animal vegetal body axis. Okay, the embryos at the animal side, there you have the divisions, it stays like that. And then at M, panel M, you see a new thing that has come up. You've seen the shield at about 50% probability. You have the dorsal shield forming, so that will be the dorsal side. And then you see the formation gastrulation of this other layer. And then you have suddenly a second body axis, which is the ventral dorsal, or we call it dorsal ventral axis. And in the end, the combination of the animal and dorsal ventral axis generates the final body axis, and that corresponds to up front here. So at the animal pole will be the front of the embryo, and here will be the end of the embryo. So the embryo will be forming on this side, this axis. And then you see this here, the embryo keeps wrapping around the yolk later on, and then suddenly the tail touches off, and then the embryo straightens out. And at 5 days of development, you already have a swimming larvae. It has a swim bladder. It swims around. It can tackle the food. It sees the paramecia. You saw these calcium influx in the optic tectum of the fish. So how they respond to visual input, and then they can actually hunt down these paramecia, which they also do with great pleasure in our lab. So the question is then, you have all these cells, you have these things happening. In the end, you have a differentiated organism with all these. Why doesn't this work? Anyway, that has all these different organs. It has a differentiated brain. It has a gut and everything it needs. So how actually does what we observe, how is that connected actually with the formation of the different organs? So we not just look at things now anymore, but we try to understand actually also some molecular mechanism and how molecules and cell behaviors sort of are integrated into this big plan of generating a vertebrate embryo. And the one question that you can ask, if you go back a slide, and that works, is what is the connection between these organs here and anything that happens up here? Do we know how these cells become specified? How do they become specified? I mean, we know already that these cells here probably are corresponding to the top of the embryo, but these cells here that form the mesoderm, where do they come from? How do they know what organ they should contribute to? and so on. So one way to tackle this is to make a fate map. So we have an embryo at 30% epiboly. You have here the cells here forming this cup over the yolk. And here you have the end result. And I labeled here in different colors, different parts of the brains, notably here the forebrain, midbrain. So this is the central nervous system. And then for mesoderm, I have here some somites and the notochord, which is the supportive structure that underlies the spinal cord. Okay, and here we have the lateral plate mesoderm. So what we talk about here is different structures from the ectoderm, different structures in the mesoderm, and how do they arise? Where do they come about? Okay, so the first thing that you can do is a fate map. And by fate map, what that means is you label individual cells here. You can use, for example, a tracer dye. You can do a transplantation of a single cell, or you can activate a certain dye in a single cell. And you do this in different examples. So every dot would be a different embryo, right? So in this case, we would have labeled 7 embryos with a single cell. And then we ask the question, where do these cells, each of the cells, where would it end up on this side? And this is, I'm just saying this now, you don't see this here. This would be the dorsal side and this would be the ventral side, okay? So we don't see any difference here, but this is how this would be interpreted later on, because you see how the embryo develops during later phases of development, and they would go, of course, through these shield stage and all of that. But you label the cells at this stage, and then you see where do they end up in the embryo. Again?
Oops.
Ah, parlez-vous français? Okay, never mind. Okay. So how do they end up? And then you find that cells here end up in the brain, in the midbrain, or they become epidermis actually. And oops, this is moving too fast here. Something went wrong, never mind. So you know when you take a cell from here, it will end up in the hindbrain. A cell here will be in the forebrain. Cells that are here are becoming somites. Cells here become notochord, and so on. Okay, so you find a correlation between the origin of a cell and what kind of structure it will become. And that is then called a fate map. If you do this many times, then you can sort of extrapolate from a single experiment, get to see a general trend, and then you see that at this stage, the embryo, there is already a fate map where sort of the plan is laid out for what is going to happen later. It does not mean that the signaling and the specification has occurred already, because the signals that specify the cell types, they can occur also later. But it's simply a correlation between, a spatial correlation between where the cells come from on what type of cell type they will become later, or which part of organ they will contribute to later. Okay, so how do cells then like this, how do they end up actually physically in these different spots in the embryo? And that is the interesting thing about gastrulation, because gastrulation is all about cell movements. And that is shown here in this video. This is basically a time-lapse video from all the stages that we looked at this morning. And I will play this a couple of times. And we've seen all these stages this morning, but we saw them separately, different embryos, short movies, so we didn't put them together conceptually as a single movie. Oops. So what happens first is you have cleavage divisions. Okay, cleavage divisions are these incomplete divisions. They are symmetric. They are synchronous. So you have go from 2 to 4 to 8 to 16 and so on cells. Okay. very fast, as I mentioned before. That goes on until about this stage, from 32 to 64. Then you have a new division plane, and you have cells that are on top of each other. So you have a second layer of cells. And as this keeps on going, you have a blastula. This is important. Blastula, during this stage, the zygotic genome becomes activated. So we have this process called maternal zygotic transition or zygotic genome activation, or also called MBT. It's all sort of different focus on the same process. MBT is mid-blastular transition that includes activation of the zygotic genome and also the shift from maternal to zygotic gene function. And this then is about, this is the high stage. This is the sphere stage. So you have a lot of cells and they sit like perfectly on top of the yolk. And then we have the first thing that happens in gastrulation is we start having epiboly. Epiboly is this formation of a cup. It involves a lot of cell movements. So the cells are becoming active and they're starting migrating over the yolk, but some of that is actually passive. They're being pushed down rather than actively migrating down, but that's a different issue. I don't want to discuss that here. Fact is, the epiboly means the cells are engulfing the yolk. Okay. At this point, this embryo is still radially symmetric. We have only one body axis. It's the animal-vegetal axis. There's nothing else that is at least evident. And then you saw the shield here. I mean, I go a little bit back. You can really catch every moment now. Here, there's a very sudden transition from radial symmetry and breaking that radial symmetry, and that is here. While here it still looks the same on this side, but this on the right-hand side, which we define as the dorsal side, you have this thick structure, which is called the embryonic shield. And you see there's a second layer forming under the outer layer. The lower layer is going to be mesoderm. It's also called the hypoblast. The outer layer is the epiblast. And as epiboly occurs, the cells are moving forward. These cells are actually moving upwards. At the same time, the cells are also elongating down here to the vegetal pole. This is what you saw very nicely in the movie this morning, I think. You have this thickening here and this thickening here, and you have some cells here. These cells will become the tail. And this is basically everything from the yolk extension up to the head. And actually here you really see now the new body axis. You have anterior to up here and posterior here. So that corresponds in general with the animal vegetal axis. And you see obviously that this will become the head and this becomes the posterior structures. This is a special, we don't discuss that today, but this also has some interesting features here. Then we have organogenesis. You see the first somites forming here. You see the differentiation in the brain. You see the otic placote later. You see that this is actually merging together. So we still have extension here. And this tail is sort of connecting here with the rest of the embryo. And voila, in 16 hours, you made a vertebrate embryo. And it all happens in front of your eyes. Okay. So this way now we can explain the body axis by cell movements. We have the anterior and posterior axis, the ventral dorsal axis, and we have cell movements of cells from this side to that side and along the body axis here, and they got different names. So the first one that we saw was epiboly, the migration of cells down from the yolk. Then we have a massing of cells on the dorsal side by migration from the ventral to the dorsal side. So this is convergence movement. And then the other layer, the mesoderm, is formed by an involution of cells under the epiblast. So if we would look at this on a fate map and we would label these different cells here, these cells would be the ones that go a little bit to the dorsal side, extend a little bit like this. This cell here, it will also go to the dorsal side and extend a little more. And the yellow cells here, they're actually forming the notochord here. They will extend all the way from dorsal to ventral. And also these cells here that are at the margin early on, they are all the cells that will become mesoderm, because all these cells, they involute. Okay, so the yellow cells here, they are the involuting cells. So these are going to be mesoderm. I should actually label them in green to be more correct. Question is, how do these cells involute? So you can also make a video of this involution. Shown here. So we have this embryo, we have this early gastra, this is a shield, there's a lot of cells moving in here, and all the way along the margin, you have involution. This involution is the formation of mesoderm. These cells express already mesodermal markers, like no-tail here, shown in red. So this is the section of the embryo. This is the epiblast, expressing OCT4.
Henry.
Yeah? Hello?
No-tail, just to mention, because I'm kind of listening, is brachyury. Isn't it? It's the brachyury or homologue of fish.
Yeah.
It's because they know brachyury quite well. So
do you?
But they should. They've heard a lot about brachyury.
Yeah, let me show.
The mesodermal gene.
They're smiling, so they don't. Yeah, good. So this brachyury, and so that is a marker for mesoderm differentiation. Okay, so early on, when these cells involute, they already sort of determined to become mesoderm. And then on the right hand, I show you this movie that shows this, how this involution takes place. Outside, this is something I have not mentioned before. This is the first differentiated cell type that's called the enveloping layer. It's really extra embryonic. It doesn't concern us in this context. It helps with the epiboly mechanistically, but what we are talking about here is the epiblast cells that here then become mesodermal. Oops, didn't work. Yeah. So you see how these cells start blabbing actually, and then they loosen up their junctions and they sort of become more rounded, and then they can move up. So they leave this group of tightly linked cells, loosen up, and then they can move forward, back up in the embryo. Time for a bit of summary. We have early phases, oogenesis, fertilization. Then we have cleavage division that lead to blastula. Then in gastrula, we have the morphogenic cell movements that form the germ layers. And during this time, these cells are already specified for the germ layers. And in the end, later on, pharyngular stages, you have the beginning of organogenesis, which means that the different brain parts are being formed, and also the somites are forming, and so on. And also the early cell positions thereby can explain, so thereby the movements also explain this fate map, because the cells here next to the yolk at the margin, they are the first cells that involute. These are the cells that are involuting, so they will become mesoderm. And the cells on top, they will become ectoderm. This is becoming the neuroectoderm, and this is the non-neural ectoderm, like the epidermis. So we know now how cell movements explain the fate map, but the question is also, how are the cells specified in a molecular fashion? Is what makes a brain cell, what makes a ventral mesoderm cell, and so on. So for that, people did this genetic analysis, and actually they found mutants early on in mutant screen, where this process that happens in the wild type, as we discussed it this morning, does not occur properly. And to make a long story short, there is 4 pathways. 2 of them are most prominent. They're called the BMP pathway, and BMP is, of course, a vertebrate homolog to DPP that you know more about now than I do, and the Wnt pathways. And what happens if you interfere with the BMP pathway is that you have changed proportions of the body. Okay, so this is an embryo, it develops normal, and in the end, you look and you have an embryo that looks like this. So I'm not expecting to answer any questions from my side now, but I think it's pretty obvious that this embryo has a real big head. Actually, it's very brainy. It's the smartest zebrafish you can imagine compared to the wild type. But the tail structures are very small. Okay. If you then go back here and look at what happens is in the wild type, this neuroectoderm consists of a different portion here, certain portion of the blastula. And now we have a lot more brain, so that it looks like some of this tissue here has been respecified into become brain, so you get a better brain in the very end. The opposite happens in certain Wnt mutants, where you have an embryo like this, and in the end you have no brain, but you have a lot of tail. Actually, there's not much left of the embryo. But this phenotype, you have a reduction of dorsal tissues, and you have more ventral tissue in proportion. So we call this a ventralized embryo. Embryos that have a lot of ventral cells. In this case, BMP, you are dorsalized because you lack ventral tissue and you have a lot of dorsal tissue. So that is something that you have to be aware about, and we get back to that later. If you didn't understand it now, maybe you get a better feeling for this later on. So again, this was the BMP, and this was the beta-catenin mutant. And this is what happened now. In a wild type, we have certain proportions that are the fate map. We have ventral mesoderm, we have dorsal mesoderm, we have neuroectoderm, non-neuronal ectoderm. In the BMP mutant, you dorsalize the embryo, so everything becomes neuroectoderm, and also the mesoderm becomes dorsalized, while in this beta-catenin mutant, everything becomes ventralized. There are no dorsal tissues anymore. So basically, there's signaling pathways that are correlated with the fate map. So actually, the fate map is a correlation, and actually the molecules that ensure that these cells are actually becoming what they're supposed to become in terms of their cell identity, that is regulated at a molecular level by a pathway such as BMP and Wnt. And today we only talk about this pathway, the BMP pathway, that is important actually for generating this fate map here at the molecular level. This doesn't need much explanation. You know, there is 2 kinds of BMP receptors. This is BMP. There's many BMPs in fish and other vertebrates. So the most prominent ligand here in our context of dorsoventral patterning is BMP2B. And you heard about SMADs and co-SMADs and phosphorylation, and then the activation of target genes. And actually, not surprisingly, when you look at BMP signaling pathway, at the bottom line, you have these target genes, and these target genes are transcription factors, and these transcription factors are important to specify as transcription factors that specify ventral fates in cells. So overexpression of these transcription factors in cells will cause a ventral fate. Ventral fate, okay? That's important. You have dorsalized embryos in the loss of function because BMP is important to induce ventral fates. And if you look at mutants in other factors like SMAD5 and other factors, then you get consistently the same phenotype. In swirl embryos, this is BMP2B. You have this dorsalized embryo. And in snail house, which is another BMP, you also have this dorsalized embryo. Sorry if I mixed that up. So these embryos are dorsalized. There's a lot of brain and no ventral tissue. And also when you have mutants in one of these SMADs, a co-SMAD, SMAD5, somatobun is the mutant, you also get dorsalized embryos. It's just brain tissue here. So how does BMP do that? So you heard about gradients. This is also about gradients, but also about general levels of BMP expression. So when we would take one of these mutants that are completely devoid of BMP, it would look like this. It's a just neuroectoderm sitting on front of the yolk. Swirl embryos are extremely BMP2B mutants are extremely dorsalized. There's nothing else. And what you can do then is, how about I inject some BMP2B RNA into these embryos? And this is what happens when you do that in different doses. At low doses, you get some rescue. And if you increase the doses of BMP2B RNA, you get almost a complete perfect embryo. Okay, so that means it's dosage dependent. Okay, that means that you gradually rescue. That means the level of BMP is important. Then you took this embryo in E, or got all the way to F. Then we say a lot helps a lot. So I do even more, get better results if I do more, better rescue. But that's not what's happening. What's happening here is that if you increase, actually you start ventralizing this embryo and you end up, it looks like a beta-catenin loss of function embryo. So you go from complete dorsalization in the loss of function to a complete ventralization in the gain of function. Okay. So that means really BMP is an instructive signal that really is able to pattern the entire dorsoventral axis of the embryo. And somehow dose is important. How can dose be important? Well, the dose is important because BMP acts in a gradient, and cells that are expressing high levels of BMP or are receiving actually a high level of BMP signal are being more ventralized than the cells on the dorsal side. Somehow they seem to receive less BMP, and this is exactly what happens in the embryo. This is an antibody against phospho-SMAD. And this would be the embryo here, as you see on the right-hand side. You have the dorsal to the right, animal ventral. And you see that SMAD, phospho-SMAD, which is the readout that we use in the lab for BMP signaling, is very high on the ventral side, but low on the dorsal side. Okay, so the question is, how can that happen? Because when we did this experiment with the RNA injection in these fish, it was ubiquitous. RNA is not selectively translated on the ventral side or dorsal side. There must be something with the distribution of the protein or its activity in some ways later on, downstream of transcription, downstream of translation. And what is peculiar, of course, is that this region that we talked about earlier, the shield here on the dorsal side, this is where BMP levels are lowest. So the idea is the shield somehow prevents BMP signaling on the dorsal side. Well, an experiment you could do is the following. You do a transplantation experiment, and this is like the Spearman experiment that was done 100 years ago. Yeah, must be about 100 years ago, 1920s, or maybe 1930s, I don't know. You take a lump of cells from here, suck it up, and stick it in another embryo on this side. So you make an embryo that has actually two shields. And this is what is done here. You suck out the shield from a donor, you transplant it on the ventral side of the host, and you label them with a fluorescent marker that you can later on detect these embryos again, or the cells that you actually did transplant. And this is what comes out of that. You make a second axis. You get a double-headed embryo with two axes. They're not complete axes. I think that is also important to know, not for you now, but in principle, that makes a difference. But especially you get two heads. You don't get two tails, you get two heads. And the head is dorsal structure. The head is on the dorsal side of the embryo, okay? That means you have generated two dorsal sides in the embryo. Okay, we know that the Spearman organizer here, it expresses genes that are able to antagonize BMP2B. Actually, caudin is an inhibitor of BMP2B. Okay, it binds to BMP and prevents signaling. Now, if you take this tissue, then of course, on the other side, you get a second Spearman organizer, and then you create a second inverted gradient of BMP2B, and that's how you get a second dorsal side and a double-handed embryo. And then it's of course important to know how is actually this activity of BMP signals regulated, of how do you generate really this gradient, and this gradient is shown here at a onset of epiboly. So this is about 40% epiboly. You see that BMP2B expression, so that's the RNA now, is on the ventral side only and not on the dorsal side. So this is a different situation from when we injected the RNA. RNA was ubiquitous, but now this expression of BMP in the end, and that's the endogenous expression of BMP, of course, is only on the ventral side. And caudin, its antagonist, is on the dorsal side. And we know that caudin directly inhibits BMP by binding to it, preventing the signaling. So we have an antagonism between BMP and caudin on the protein level. Okay. Another question is, when is, never mind this Bosozoc thing here, when is BMP being expressed? Okay, if you do an in situ, RNA in situ, and you look at a very early embryo, right after cleavage stage, so this is a very early blastula, 128 cells. So that is, what is that? It's like 6 cell divisions or so. This is not in situ, I just pasted it in, but you would not detect any BMP. Okay, so that is interesting. So it's not maternally contributed. Okay, the embryo has a lot of proteins, it has a lot of RNAs, but it does not have BMP2. However, only 1 cell division later, you see a very strong staining for BMP2, and it is ubiquitous. And this is basically the situation that we generated when we do this rescue experiment, when we inject the BMP RNA into these mutant embryos in order to rescue the phenotype. And this explains why we get such a good rescue, because even endogenously, the protein or an RNA is expressed ubiquitously in the embryo. However, and this is just a bit on the side, at the same time, this thing here, it's an inhibitor of BMP on the transcriptional level here. It's called bosozoc. Don't have to worry about that, what that is turned on, and only one cell division later, so like half an hour later, you see that the area where bosozoc is expressed is lacking BMP. So bosozoc is a repressor of BMP expression here. And this is basically how the cascade of generating this gradient of BMP is started, okay? So you go from ubiquitous expression, you get from no expression during early stages to blastular, you have expression of BMP2B ubiquitously, but then you get ventral restriction because it is becoming repressed at the dorsal side. Then you set up this gradient, and then this gradient can act during gastrula and pattern all the cells during embryogenesis.
Henry?
Yes?
Can you go back one slide?
Yeah.
What do you see here? There's no ventral expression of anything.
Oh, this is just the embryonic stages.
The black
this is
color?
Just to show, this was just to show them the stages. This was already, the data was shown here.
Okay, because it's... Yeah.
This was basically a summary slide.
Okay, yeah. The black doesn't mean anything.
No, that's just a shadow from the illumination.
Okay.
Okay, this we saw already earlier. Okay. And this is now a little bit on the side. There's a few more slides to come. I just want to point out that in contrast to mammals, maternal contribution is a major issue in embryos, invertebrate species that have a lot of yolk. So the yolk provides a lot of nutrients and stuff like that. And the first axis, animal vegetal axis, is already early on specified, actually during oogenesis. And the early phase, when the animal vegetal axis is made during egg maturation, a lot of material is planted into the future animal side, vegetal side of the embryo. So what we deal with then early on, up to mid-blastular transition, is a complete maternal genetic contribution to embryonic development. Everything is provided by the mother. Okay. Then, after this MBT, the dorsoventral axis is specified, and on top of this animal vegetal axis, you start having this dorsoventral axis, which is becoming pattern, and then you end up on a complete zygotic program. You end up with a patterned embryo. I skip this. Okay.
Why do you skip it, Henry? It's beautiful.
Yeah, okay, I show it. I don't remember what I wanted to say
just
here.
Run it.
Okay. So, okay, what I want to say here is the maternal contribution is extremely important for these early cleavage divisions, okay? As mentioned, every 20 to 30 minutes, you have these divisions. They are synchronous, and you can actually look at these cells by just injecting nuclear GFP, and then you can follow in 3D every single cell, how often it divides and how it migrates. And so this was a study. This is really nice. I mean, it's done on a SPIM. You see actually here that the cell divisions are really synchronous in the beginning. On the left hand is an animal view. This is a side view, a vegetal view, and you see during epiboly, these cells how are they going to the vaginal pole? You have this closure of the yolk plug, and then you have formation of the body axis. And actually, you can compute the behavior of every single cell during these time-lapse movies. Maybe I show this again. And there's not much happening anymore.
Yeah, you see the somites now.
Or not anymore. So you start with the cell. So this is the animal view on the left. This is the view from the top of the embryo. And the one hand on the right-hand side, you see this embryo from the opposite view. So the cells are moving towards you from the vaginal side. And you see this pulsing of divisions in the beginning. So that was when it was still maternally controlled. You have these cells here, they're called the forerunner cells. So I don't know exactly, they have some biological significance, but I don't know exactly what they do. So this is the end of puberty. So actually during puberty, of course, you have already formation of the body axis. So this would be the dorsal side. This is where the head is forming. And this is where the tail is forming. And now we wait for the somites. It must be somewhere on the right-hand side. Yeah, you see some stuff.
It will come.
There's so much use. Yeah, could be. It's kind of funny because they don't look like they forming in a posterior to anterior direction. Anyway, I'm not an expert at all on these maternal zygotic transition. I think Alex had a last year paper on a gene called NANOC, which is one of these stem cell factors. But just to give you an overview about what's happening and why I think also this is interesting is that, of course, in these embryos, you have all these maternal transcripts and you have these very, very fast cell divisions on the bottom. So you have S-phase mitosis, S-phase mitosis, okay? And then after 1K, so 10 cell cycles, it becomes, it slows down, it becomes asynchronous, and actually you have a longer G2 phase. And I don't know these things anymore at the top of my head, but what's interesting here is that the cells here are in a way highly specialized. And then there's a transient phase of totipotency. And that is kind of interesting. I don't know the details anymore, but it is the interface between the onset of zygotic gene transcription and the complete takeover of zygotic transcripts. And this transition, mid-blastoid transition or maternal zygotic transition, there are three key events. One is that you need to time the zygotic gene activation, the activation of the zygotic genome itself, and also the removal of maternal transcripts. And all these things are highly regulated. I don't go into any, Keynote quit unexpectedly. So I reopen. Are you still with me? Okay. Open, recent. Okay.
You have to share screen.
Yeah. Oh, okay. Yeah. Okay. Okay. So there's active mechanical. No, that is a. Okay, okay, let me. So this is new. So keynote. Okay. Okay, I just go to prevent any more delay. Share. Okay, so I just wanted to let you know that it's okay now, right?
Yep.
No?
Seeing is believing.
Yeah, now we see.
Oh, you see? Okay. Okay, good. Okay, so, and this maternal zygotic activation is important for any development. So if you prevent, for example, zygotic transcription by inhibiting RNA polymerase using this drug here called alpha amanitin, you don't have zygotic gene transcription, you don't get any development anymore. And the same is true if you inhibit any of these stem cell factors like NANOC or OCT4 in zebrafish. You prevent activation of the zygotic genome, and especially, and this is how the circle is closing again, in an OCT4, this is OCT4 mutant, you do not get expression of BMP2B anymore. That shows that the stem cell factors in the end are the factors that actually turn on the developmental program in this context. And you need OCT4 to kick off BMP2 expression at MBT, and that allows then activation at MBT, and then the formation of the gradient, etc. And this is it for now. So at, what does the program say? Does it say 2 o'clock?
Oh.
7 o'clock. So this is the afternoon now. I went a little bit over time. We do this part, seeing is believing. We stick together to the same groups. Alessandro, you inviting me to the meeting later on?
Yes, I will.
This will be done in several rounds. We have several rounds of movies, which we'll discuss. No, first we go back. Daniel, you will give an introduction, right? Or should we go into our groups later on? Daniel?
I think we can go directly to the groups and then we can tell them there the steps and we will go through.
So we meet in the groups and then discuss the procedures. Yes?
What about the introduction? So
yeah, we do the introduction in the groups.
Oh, okay.
Yeah.
But not Daniel.
Well, there is not much.
Daniel does it. We do it all, everybody in their own group.
Okay.
Like this morning, basically. So we do 4 rounds of analysis that we look at these videos, then we discuss the videos, and that is actually copy paste. See what I wrote here from the program. So we do home office. We take a 15-minute break then. So we meet at 2:45. Yeah. So don't be scared. We catch you later. Okay, so I stopped the screen share. And Okay.